Today we are going to talk about dark genes and young genes. Well, yesterday in my first lecture I explained about the gene structure, what is basically a gene. I once again repeat that the gene is a unit of a heredity which is basically passed from parent to a child. And as I told you that all the chromosomes which are present in our nucleus or in a cell are made up of DNA. And again I am showing the diagram in which the first diagram you can see a strand consists of a centromere. There is a one short arm and another is a small arm. So this is a single chromosome. At the time of a division, the chromosome become bivalent. Bivalent means there are two strands. So in the next diagram you can see there are two strands and the center portion is known as a centromere or a kinetochore, and the parental chromatids and the sister chromatids means the two chromatids are formed or two strands are formed here. Well, as I told you that the DNA is basically present inside the chromosome, and genes are also located on the DNA, or it is basically a part of a DNA sequence. So in the another diagram you can see in the center I have written gene 1 and gene 2. So this is the first gene, then there is a small space and there is a gene 2. What are basically the dark genes and young genes? Well, it was actually discovered long back, and there was a paper in Nature where researchers showed that the gene may function as a switch for human diseases. Large number of human diseases are reported, like multiple sclerosis, which is a muscle disease, lupus erythematosus, rheumatoid arthritis, Crohn's disease and celiac disease. And these genes may also function as a switch for certain specific traits like height, color of eye and some other things. Scientists found that minor changes in human DNA sequences increase the risk that a person will get these diseases. But those changes were found in an area which is that time called as a junk, but now it is referred as the dark sequences. But their significance is still not clear very well. Still, the researchers are underway to discover that what these dark sequences are there in the DNA sequence. Analysis reveals that many of the changes alter gene switches. These changes are highly significant in terms of the development of a disease. For example, there is a study in the case of a cancer. When the researchers were determining the sequences of cancer cells, they realized that most of the DNA changes in cancer cells were not in basically the genes, but they were in the area which are nearby genes and now referred as the dark regions. But the challenge was to figure out which of those changes are in these dark regions are basically responsible for the growth of a cancer. A similar situation was found in the case of a prostate cancer, where it was found that a change in a gene are not readily attacked by the drugs. What does the dark genome or dark gene mean basically? Well, if you see the structure of a dark genome or a dark gene, it is basically a concept which involves two regions. One is known as a non-coding region and coding region. But basically, the non-coding regions are basically called as a dark genome. What is the meaning of a non-coding and coding regions? I will explain later. Well, if you see in the terms of a function of a gene, then in that case the non-coding genomes are those which are basically not related to the function of a gene to form or to synthesize the protein sequence. But they function as a regulatory proteins, regulatory regions, and basically they regulate the gene expression. The regulatory genome or regulatory genes regions include large number of nucleotide sequences which are basically transcribed in the cell, or transcribed means they are interpreted in another sequence which is basically known as a non-coding RNA. I already told you in my first lecture that the RNA always formed on a DNA by a process called transcription. In these regions, some special elements are there. Elements means element I mean to say is the small sequence. They are known as transposable elements. Well, these sequences were first time identified by Dr. Barbara McClintock, and they are very interesting. If you have seen the maize, you can see there are a large number of colors in the maize kernel. Like yellow is there, dark brown is there, sometimes there is a black is there, and in the one fruit you can find all these colors. So Barbara McClintock was interested to know that why these type of different colors are present there. And then she suggested that there are certain genes which jump from one place to another place. And such genes which can jump from one place to another place and then express their characteristics are known as the transposable genes. Well, as I told you that genome is basically divided into coding genome and non-coding genome. So coding genome are basically those genes. There are about 23,000 genes are identified in the coding region, and they are all synthesizing proteins. And non-coding genome, that I already told you, that they do not produce the proteins. Over the last decade, it was discovered that about 98% of the human genome does not code for the protein. So this is all comes under the non-coding genome region in the DNA. Although non-coding genome regions do not provide instructions for making proteins, but they are actually very important for the hereditary informations. Heredity means the characters which are passed from one generation to the another generation. Now, I discussed about the exons and introns. In this diagram, I have shown you, coding regions and non-coding regions. Now, you can see clearly here in this, there is a DNA sequence. The greens regions are basically the coding regions, and in molecular terms, they are known as exons. And the yellow regions in between are known as introns, which are basically non-coding regions in which we are interested basically. This is a diagram based on Human Genome Project, where the different genes are identified in the human genome, and you can see that the introns are 26%, miscellaneous unique sequences 12, 8%, 5%, 3%, 3%, like this. The protein coding genes are only 2%. That means, as I said, 98% of the human genome consists of non-coding regions. These are the diagrams to explain the non-coding RNA which is formed and the transposable elements. And just a comparison of the coding regions and non-coding regions, and if you want to know it, they have coding regions have a DNA which encode for proteins. The coding sequences has enzymes. They are about 1% of the total genome, and they encode proteins which are regulatory in function. Non-coding regions have DNA sequences do not code for proteins. They are regulatory elements. They are in the form of introns. Repeating sequences means one sequence is there and another the same sequence repeats in a DNA. Pseudogenes which are present but have no function at all, and people still don't know what are they. And then the telomerase, as I said in the earlier lecture, the telomerase are the last end of the chromatid or chromosome strand. So, and they are basically, as I said, 99 or 98% of the formed by the non-coding regions, and the main characteristic, they control or regulate the gene activity. Just to explain that how the protein is formed, I already explained, but just to repeat it, DNA is there, DNA undergo transcription, form RNA, which is very important component of a protein synthesis. Once the RNA is formed, basically the information for synthesizing a special protein, suppose like a myoglobin, which is a muscle protein, or some other protein which is used in the heart muscles, or protein used in the brain, protein used in the skin, or everywhere. Whatever protein is synthesized is basically the information of that characteristic of that particular protein lies in a DNA, which is basically transcribed it in form of RNA. And since RNA has the information which in the form of a nucleotides, so ultimately it is translated in form of a protein. This is basically the process. exon, intron, exon, intron, exon, intron. You can see in the gene DNA, a particular gene consists of large number of exons and introns, and there is a small sequence which is light blue known as a promoter. This is basically a regulatory region. The remaining part is known as a structure gene, and this is known as a regulatory gene. This promoter part basically controls the entire regulatory, entire structure gene. Exon one, first exon may be responsible for some particular protein. Exon 2 may be responsible for some other protein. Exon 3 may be responsible for other protein, and exon 4 responsible for other protein. So simultaneously, the strand for these different type of proteins can be transcribed on a single DNA, or the different part of a particular protein can be synthesized due to presence of sequences located on the different locations. It has also been described that there are certain animal genomes which are completely missing certain genes. Those might have appeared long back in the similar species and must have present to keep those animals alive. But those animals are not present on the earth. These missing genes are also dubbed as a dark DNA or dubbed dark genes. In case of multicellular eukaryotes, means the animals which have more than one cell. Bacteria, viruses, yeast, they all have a single cellular structure. But other animals higher to it consist of 2 cells or maybe thousands of cells, or where a person like us where more than enormous number of cells, which is very difficult to count. So large number of cells are present. These are called as a multicellular eukaryotes or multicellular animals. They also comprised of a DNA which does not encode for the proteins. So this kind of a dark DNA is present right from the single cellular animal to the multicellular animals, including primates and humans. But we still do not know why it is present. The so-called dark DNA is believed to consist of highly mutated hotspots that are causing the rapid evolution. When there is a mutation where there is something change in a DNA sequence, new combinations are formed, and because of that, basically it is responsible for the evolutionary kind of changes. Large number of such type of dark genes are actually identified as well in the last decade. One is known as a SLX41P, which is involved in glucose metabolism in our body. Another is associated with coronary artery disease, and there is also one which is associated with attention deficient hyperactivity order, which is basically found in the case of children. What is basically perspective of the dark genes? In spite we know that they are not synthesizing the proteins. Well, it is very interesting. This particular area, I cannot go into details because of a time limitation, but basically these areas provide the therapeutic opportunities for developing different drugs to certain diseases. It can also help to search for genetic variation, which is basically responsible for the evolution of the animal species. But still these dark genomes may generate certain proteins. They are vital for brain development and may be responsible for increasing the person's risk for developing the diseases like schizophrenia or bipolar disorder. The first time this type of dark gene was studied in an animal called a sand rat. The research found that in this particular animal there are large number of mutations in the dark gene area, and it was found very interestingly that basically the change was, or the mutation was found in the area where the GC base content was much more. GC means guanine and cytosine. The diagram is enclosed here of the sand rat. So these desert set, due to this type of a mutation, might have gone under a fast evolutionary jump and fast evolutionary jump in their species. If there is an excessive mutation of this kind, this type of can often stop gene from working. A particular gene can be stopped from working or being undergo the silence, so it will not produce anything. In addition to desert rat, dark DNA was also found in the case of a DNA, also found in the case of a bird. Today, as human-driven climate change is also speeding up evolutionary process among many species, more cases of dark DNA could result. A study published in Nature Genetics found that mutations outside of gene coding regions can cause sometimes autism in the children's. There is also a statement by certain scientists, and they believe that the bacterial genomes show lower gene density as comparison to the multicellular animals, but that lower gene density than average due to the accumulation of different type of sequences that are similar to the eukaryotic genomes. Sometimes earlier they were considered as a dark or a junk DNA. It is also believed that some of the gene sequences once considered junk are actually fragments of viruses left buried in our DNA from an infection in a long forgotten ancestor. Means long back some bacteria, some viruses might have entered our body, and then once they died, they left their DNA, non-functional DNA in our body, and these DNA ultimately evolutionary process incorporated into our DNA sequence, and now since they are not working or they are not producing any protein or they are silent, that's why they are now considered as a junk DNA. Thank you very much. We will talk about the next lecture. In this lecture, which is the third part of the lecture 7, I am going to talk about the genes and their role in with hormones on behavior. So, gene, hormones and behavior is the basic title of our this lecture. Well, there is a common question in biology. Are hormone response in body are controlled by our genetics? First, let us understand what is hormone, and then we will talk about to answer this particular question. Hormones are basically the protein, mostly protein substances, which are synthesized from our glands. You might have heard about the names of different glands, like pituitary gland, which is present in our below our brain. There is a thyroid gland, which is present in our neck. Just above the thyroid, behind the thyroid, there is another gland which is known as the thymus. Then there is a gland just above the kidneys, yellow in color, you might have seen in the photographs, known as the adrenal gland. And in addition to that, there are a large number of genes also associated with the testis and our ovary. All of these glands are basically responsible for secretion of a hormone. I will give you a few examples to understand it. In case of a pituitary gland, large number of hormones are formed. This gland is divided into two parts, and one is called as adenohypophysis, and the smaller part is known as a neurohypophysis. Like neurohypophysis area is responsible for production of oxytocin. Adenohypophysis is responsible for a hormone called as a growth hormone. If the growth hormone is not present, our growth will be stopped. Okay. So, these are similarly like you might have heard about the TSH, thyroid stimulating hormone or thyrotropic hormone. These hormones are basically responsible for our metabolism, and they are synthesized from the gland called as thyroid. The thymus is also responsible for another hormone, thymine, and these hormones are basically, or this particular gland is related to the development of certain cells which are fighting the bacteria, viruses which enters into our body, or a toxic substance which enters into our body and try to develop a disease in us or develop pathology in our body. So, these hormones, adrenal gland produce hormones which are responsible to control the functions of the water absorption and ion absorption, like sodium ion is absorbed from the urine in our body. So, hormones have specific functions. Most of the hormones are protein structures. So, when they are protein structure, they must be having their single unit known as a amino acid. So, all hormones are made of amino acid because proteins are made up of amino acid. And here I tell you that as I said earlier, that each gene or DNA sequence consists of 4 different types of nucleotides which are arranged in a repeating sequences, and this DNA consists of information which type of hormone has to be synthesized in a cell. Suppose growth hormone need to be synthesized in a cell. So, the sequence or gene sequence of these nitrogenous bases will be present at a particular locus in a DNA that will be transcribed to RNA. So, the growth hormone information will be transferred to RNA, and then this RNA will ultimately responsible for arranging the different amino acids which should be present in growth hormone. So, finally, the protein is formed which is actually the growth hormone. So, in this way our cell functions and produce the growth hormones or general hormones. The similar phenomena occurs in almost all glands. Now, another question here, how much do genes determine a behavioral trait? Well, simple answer to this question is that both genetics and environmental factors play a major role in determining our behavioral traits. Hormones are mainly proteins secreted from endocrine glands like pituitary, adrenal, as I already told you, and they play important roles in development, differentiation, in puberty, influence events like sleep cycle, urine formation, cardiac cycle, and to name a few. Hormones are also important factors that help our bodies to respond to the environmental signals. When there is an environment change, your body immediately responds in form of secretion of hormones. Our genes do influence hormone secretion. It is a definite statement. And there are certain examples. I will take the example of a growth hormone which I already took in the initial phases. For example, the gene for growth hormone is known as a GH1. In case of mutation in this particular gene, it may not be able to form or synthesize the correct amount of a GH required for normal development, or it may not be able, since the sequence is completely changed by a mutation, it may not be able to produce actually the growth hormone, and instead it may produce certain else which may not act as a GH. If a GH is produced in a low amount or less amount, in that case it affects the age and also affect the height of a person. So, basically he or she will be always shorter than the typical height. How growth hormone act on a cell to affect these characters? Basically, every cell consists of certain receptors. These are protein locations which bind with the hormone, known as receptors. So, every hormone which is produced by different glands have its own receptor on different cells. Like growth hormone has a receptor, and this will be called as a growth hormone receptor, located on a cells and basically respond when once the growth hormone binds, it will respond to the growth hormone. Well, if the GHR growth hormone receptor gene is mutated, initially I said growth hormone gene is mutated. Now, if I am talking about the growth hormone receptor gene is mutated, then although since the growth hormone gene is normal, growth hormone will be synthesized, it will be produced in the equal amount which is required in the body, but basically since the receptor is mutated, its structure is changed, so it will not be able to bind with the growth hormone, and in other words, we can also say that basically the growth hormone receptor will not respond to the amount of growth hormone which is available in the body. But the system is not so simple here. There is again some complications. Growth hormone gene and growth hormone receptor gene are not only the genes that affect the growth. In fact, when growth hormone message is received by its receptor, an entirely different gene which is known as IGF-1, which is basically insulin-like growth factor, becomes active or becomes turned on. This hormone IGF, insulin-like growth factor, is actually responsible for our growth or for the development of our height of the body. If this gene also undergoes mutation, then what happens? The body can sense presence of a growth hormone in the cell, but it can't respond to it, and person will still be shorter than the having a typical height. Things can get more complicated due to another gene related to secretion of a growth hormone, and this is basically a gene which is known as growth hormone releasing hormone gene. This growth hormone releasing hormone gene or growth hormone releasing hormone is also required before the release of a growth hormone from pituitary, because this GHRH GHRH hormone basically stimulates the release of a growth hormone from the pituitary. And this hormone is also under control of a particular gene which is known as a GHR gene. So if suppose this happens, that there is a mutation, in that case the body, when to release the growth hormone, it will not be able to recognize and will not be able to, or will not be able to sense, or will not be able to release the growth hormone in the cell. How many genes are basically involved in one function? Can you see that in the case of a female estrogen hormone, which is responsible for all female functions, about 80 genes are playing important role. Only then this female hormone can function. Just the missing function of a one gene or altered function of one gene can totally change the effect of a estrogen hormone. Genes matter much in hormonal action, but environmental factors actually play a bigger role in comparison to the genes in shaping day-to-day hormone responses. In fact, our hormonal responses are largely guided by cues available from our environment, and this can be explained on basis of certain examples. For example, food which we eat provides us energy and also building blocks for like protein, carbohydrate and lipids, which our bodies needs to function and grow. If enough food is not available to body, our bodies make sure that the limited energy is used to keep our bodies functioning, and the body will stop responding to growth hormone and growth will completely stop. There is another hormone known as a melatonin, a hormone secreted from pineal gland, which is located on the top of a cerebrum or cerebellum, sorry. This pineal gland hormone, which is called as a melatonin, is actually responsible for our sleep-wake cycle. This cycle or any other character, like we sleep at a particular time, we wake up at a particular time, then again we sleep at a particular time, we feel thirsty at a particular time, and then again we feel what you call to eat food. So these timings are actually synchronized by the hormone called as a melatonin. This type of phenomena, a cycle of this kind, like a sleep-wake cycle, are also known as the circadian rhythms of our body. At night, body produces lots of melatonin hormone, which causes some changes in brain activity that help to fall asleep. But during daytime, the level of melatonin decreases, so we feel less sleepy and becomes more awake. So this is an example basically that how a hormone gene is basically controlled by the environmental changing conditions, light and dark cycle. Melatonin levels increased after sunset and then decreased at sunrise, creating a consistent sleep cycle. But now, as we have light bulbs at our home or street, that keeps our environment brightly lit even after the sunset. And under that condition, some scientists believe or started investigating whether this impacts melatonin levels in sleep quality, because large number of people these days say that they are sleepless and they are not able to sleep properly. So basically the 8-hour sleep which we required is not possible. Though a lot of work still has to be done to fully understand the mechanism, studies do suggest that exposure to these light conditions for a long night can delay the sleep. And these are a couple of examples which show that many non-genetic factors impact our hormonal responses. Aging, diet, stress, sleep deprivation, pregnancy conditions, immune responses and medications can all cause levels of certain hormones to go up and down. Very briefly, we will discuss, does hormone can also turn genes on and off? And does this affect our behavior? Well, best example is of estrogen hormone called as a female hormone. Estrogen affects the expression of hundreds of, maybe even thousands of genes. And these genes may have many different functions, playing roles in formation and strengthen bones, heart functions and development of a cancer. There is another hormone known as a cortisol. When you are under stress, cortisol is synthesized in our body and create anxiety and certain other conditions, which is known as a flight and run response. This cortisol hormone is called as a stress hormone. In small animals or the other mammals, it is a corticoid hormone, but in humans, a cortisol is produced. The receptor that recognizes cortisol is found on different type of cells throughout the body. When this receptor is activated, it changes the expression of thousands of genes. These genes are about 10 to 20% of our genomes. These genes, once cortisol is produced, do you know that our immune system is compromised, metabolism is slowed down, growth is also slowed down, and many more processes based on what type of cell is expressing them are affected. Cortisol level rise during stress and thus sometimes found to be associated with the negative effects. As I said, the anxiety and increase in heart rate, etc. It is possible to demonstrate both direct and indirect actions of genes on mammalian social behaviors. Direct effects of estrogen receptor gene expression and progesterone hormone receptor gene expression prominently in well analyzed neuroendocrine mechanisms for sex behavior. Among indirect effects, the consequences of sexual differentiation and display of complex dependencies in case of male and female in various species are the basic examples. In several papers published in Cell, Nature or Science journals, researchers working with Drosophila reported that targeted expression of particular gene or deletion of certain genes can markedly alter the Drosophila behavior. So, if you remove the particular gene from an animal or knock out a gene from a particular animal, the behavior related to that particular gene will be altered or completely stopped. Is there a possibility of gene change naturally? One need to indulge in some sport. Yes, it's possible. One need to indulge in some sport, physical activities like dancing or running on a regular basis, and then in that particular conditions, the body will thus activate the genes needed to support these particular activities. So, it is possible that genes may get changed naturally. Thank you. In this lecture, follow-up lecture, we are going to talk about the genes and body features like color, height, general structure and how they are controlled by the gene function. Well, if we talk about these kind of characters, there are two words are used basically in genetics. One is known as a genotype and another known as a phenotype. Genotype means the total genome which is present in a particular nucleus of a particular species. Like in our case, you might know that in our nucleus, the total chromosomes present are 46. Okay. Similarly, there is a specific number of a chromosome in the case of a rat, which is a different species. Similarly, there is a specific number of chromosomes in the case of a giraffe, and similarly, there is a specific chromosome number in the case of a any other animal, maybe a small animal like you can call as a fish or a hemidactylus or anything. So, that means each and every animal has a total genomic content in the nucleus which is well defined for that particular species, like for humans, for rat, for giraffe, for fish or for rat. So, basically the genotype is genomic content of a particular species. Phenotype is basically the characteristic which are physical characteristics, and that is why they are known as phenotypic traits. Phenotypic traits are of two types. One is qualitative and another is quantitative. The qualitative traits are basically classical traits are known as Mendel's traits. For example, forms means round or wrinkled pea seeds. For example, horn or hornless condition in a cattle, which is structural qualitative test, and pigments like black or white coat of a rat, guinea pig, etc. These traits are always under control of two or more alleles of a single gene. Now, what is allele? I told you a gene is present on a DNA, and that particular gene is responsible for a particular character like a white coat of a rat or white or the hornless condition of a cattle. For this, there are two genes on a DNA. They are located side by side normally. they are also, we can also say that this is a pair condition of two genes. When a similar gene is present on the same locus on a chromosome or on a DNA, these genes are known as alleles. Okay? So, two or more alleles of a single gene are basically controlling a particular trait of this kind. Quantitative traits are measurable phenotypic traits. For example, a height of a man or height of a cow, weight of a man or weight of a cow, our shape, shape of any other animal, pigmentation on a skin. Some people have a very light skin, some people have a dark skin. So, it is basically the accumulation of pigmentation controlled under genes. And similarly, the amount of flowers on plants, amount of fruits on plants, seeds produced by a particular animal, a particular plant, amount of milk produced by a particular cow or goat, meat, total meat of that particular animal, number of eggs produced or total milk production. So, any characteristic which is related to the basically quantity are basically known as a quantitative phenotypic traits. In contrast to qualitative traits, the quantitative traits may be modified by the environmental conditions, and they are always governed by many genes, about 10 to 100 genes. A very classical example is there. In long, maybe hundreds of crore of years ago, the giraffe might have the neck exactly similar to the donkey or exactly similar to a horse. But under environmental conditions and when the environmental changes, the food was not available. Slowly and slowly, the lower shrubs and bushes were gone, completely disappeared from the earth, and then only there are the tall trees, and the animal giraffe has to try to reach to those tall trees, and slowly his neck is increased into such a length that it can easily reach to that particular plant or the food which is on the plant or on the trees. So, in this way the environmental conditions or natural conditions changes the or affect the quantitative traits. Genes which do not have alleles but affect a phenotype of a quantitative trait is called basically a polygene or a cumulative gene. If there are many genes of this kind for one quantitative trait, they are named as multiple factors. How these traits are controlled by genes, there are certain examples which you can understand here. This is the first example, which is a seed which is known as a kernel. It's a red in color and a white in color. So, suppose if we take these two seeds, one is red in color and another is white in color, and if we see the genotype, means they will consist of genes like R1, R1, R2, R2. अब here R1 and R1, R2 and R2 both are alleles, and they are basically dominant character. Dominant character means they will always appear in so many generations. Maybe one generation, two generation, three generation, fourth generation, fifth generation. This is a dominant character, so this will always prevail. Okay, so R1, R1, R2, R2 are responsible for the red color, while the small r1, small r1, small r2, small r2 are responsible for the white color of a kernel. If we cross between these two varieties, in the first cross, it is always known as first generation. Okay, so the genotype of the first generation will be always light red, because although the red is dominant, but white will have some influence because the genes are there. Therefore, R1 red, R1 small r1 white, capital R2 is red and small r2 is actually white. So, both type of genes are present here. It was not present in the case of a parent. So, combination is different, you can see here. So, both the color will appear, but the red is prominent. So, light red color will appear in the first generation. Now, what we can do is to check this, that the similar conditions occurs in the next generation. So, if we suppose cross between this light red condition with another light red condition having the same genotypes, see what happens in the case of a F2 generation. Results are shown in this table. So many genotypes and their ratio is shown, a number of contributing alleles, how many genes are basically responsible for a particular color, and the phenotype which is prevailed or the color which is prevailed, and we are not talking about the phenotypic ratio, it is not that important. See, in the first case, if we do the combination or we multiply it, then see how many combinations are possible of the genes. In the first block, you can see R1, R1, R2, R2 will be repeated. But the genotypic ratio is 1. Number of contributing alleles are basically 4. And since all the dominant characters are present or dominant genes are present, the child will be, or the next generation will be the dark red. Then see in the next case, there are two combinations, R1, R1, R2 and then small r2, or R1, small r1, R2 and R2. This is another combination. In both the cases, two alleles are basically responsible, and three alleles are basically responsible, R1, R1 and R2, and the color will be medium because there is a combination of the dominant character as well as the white character. See, in the third case, there are four combinations possible, R1, small r1, R2 and R2, and in the other combination you can yourself read out and see this is a combination which has the light red color basically. Okay, and in the next block you can see here again the genotype R1 capital, R1 small, R2 small, R2 small. Well, here it is very interesting. R1, one gene is there. The remaining are alleles of the recessive character or small character, R1, R2, R2. And that's why the red color will be little down, and now here you will see very light red because white will be prominent in this particular case. And the last one, R1, R1 both are small, R2, R2 both are small. So basically since the recessive gene is present here, so this will be the totally white. The ratio total if you see is 1:4:6:4:1. You may prepare any type of combinations, any type of gene combinations based on this example. The same phenotypic ratio will always occur in the case of a F2 generation. This type of diagram shows how particular characters, dominant or recessive characters basically inherit from one generation to the next generation and finally to the next generation. Let us see another example, which is a skin color in men. This example or this kind of a study was first time done by a Davenport in 1913, and he actually tried to use the pairs of a gene. One is A, small a, capital B, small b. A is a dominant character and small a is a recessive character. Okay. Similarly, the B is a dominant character and small b is a recessive character. These two genes are basically for the skin color or skin pigmentation. And these two genes basically shows the Negro condition, means very dark skin condition, and Caucasian people like us, which are not basically white, but basically a light dark brown condition. These genes were found to affect the character in an additive function, means one gene supports another to express. If a person has a perfectly dark color skin like the Negroes, the combination of gene will be AA, BB, because A is a dominant and B is also a dominant. And so the allele condition is AA and BB. In the case of another case where the skin color is light or Caucasian, there the gene combination will be small a, and the small b. This will be a recessive character. Why we call it recessive? Because when there is a dominant character, it will always subside below the dominant character. In F1 offspring, after mating, AABB and AABB, all offsprings will have an intermediate character. Not very dark, not very light, as intermediate color of a skin, and this color is called as a mulatto, which was first time called as mulatto by Davenport. Here is an example you can see in this cross. The parent's condition is always known as a P1. You can say there is a cross between AABB, which is a capital Negro and dominant character, and AABB, which is small and white character or a small white skin, we can say it. And when we see in the F1 offspring where the intermediate mulatto color appears, all the children's will have the combination of gene alleles capital A, small a, capital B, small b combinations. Now, if we have an intercross between these two characters or between these two children, we will have in F2 generation this kind of a expression of these genes. One child will be completely black Negro and it will have a gene combination capital A, capital A, capital B, capital B. The another will be dark but not exactly black because it has a combination capital A, small a, capital B, capital B. Because small a has come here, the dark color is little minimized. Similarly, you can see here capital A, capital B and small b. So, again the character is minimized. Okay. In the third case, intermediate color and see the combinations, you can very well understand by my explanation the capital A, small a, capital B, small b, and similarly the other characters. So, in this case the coloration of all the childs will be mulatto. The next case will be light like capital A, small a and small b, small b. So, this combination will make it a light color, and then finally there is one child which will have a completely recessive character capital A, small a, capital B, small b. So, this is a situation which exists in the case of these animals. Sorry, I am sorry, it should be the last white will be AA, not the capital A. Okay, very briefly we can also talk about the eye color in man. Have you ever observed that when a child is born, its color is basically light brown in color? It's not definitely a dark black or a blue color. It's normally it is a brown color. Slowly it changes and ultimately become the black color in most of the cases, or otherwise it is the blue color. Mostly the four colors prevail in the eye. One is a brown color, green color, dark black color and the blue color. These colors are basically responsible with the gene, because genes produce this particular type of a protein which is responsible for these colors, like a brown protein, green protein, blue protein and the dark black protein. And it is said that the genes are linked to a X chromosome. Well, here I have to explain in brief that if we see the chromosomes present in our cell, as I said, there are 46 chromosomes. But 46 chromosomes are double. Actually, it is a 2x number. Okay, is a diploid number. It is called. Means actual number of a chromosome in our cell is 23. Out of 23, 21 are autosomes. Autosomes means they are responsible for other characters like body shape, body color, body height, body weight, some other characters. But the two remaining out of 23 are basically known as a X and Y chromosome. Y chromosome is known as a male chromosome. If anywhere the Y chromosome is present, then the child will be always male. But if the X is in combination with the X and Y is absent totally, in that case the child will be always female. So, very interestingly, the eye color of a man is always associated with the X chromosome. Okay. In order of increasing amount of melanin pigmentation, these eye colors can be defined as a light blue, medium blue, dark blue, grey green, hazel color, light brown, medium brown and dark brown. So many colors, about 9 different type of colors are possible in the eye, and number the contributing alleles for these colors has been shown in the table here. If you see the light blue color, only single gene is present. Medium blue, again single gene is present. Dark blue, 2 genes are responsible, which are alleles. Gray color, 3 genes are responsible. Green color, 4 genes are responsible. Hazel, 5 genes are responsible. Light brown, 6 genes are responsible. And medium brown, 7. And finally the dark brown, 8 genes are responsible. So, number of contributing alleles are shown in this diagram against the eye color. Thank you very much.